but the giant oak tree in my yard is coming down today, and the tree guys are up in the tree. Yeah, and almost everybody else is gone. So if I hear the doorbell or buzzer, I'm gonna have to go and just see what's okay. But they promised that they would not fall out of the tree and kill themselves. Anyways, all right. I think everybody has record permission who might want it. We have a couple things to talk about today, and we're going to start with lab lecture because that's the fun part. Can you hear the chainsaw? I'm just checking. Anybody hear that? No? Okay. Well, every now and then you'll see me go, like that, and we'll see what happens. Also, this morning, mysteriously, during PlanFids, my internet stopped. Fortunately, Susan was co-hosting, and she just kept things going. We have no explanation for why the internet went out. The tree guys are not here yet, so it's not their fault. If that happens again during this class, that my internet goes out, the Zoom session will end, but as soon as my internet comes back on again, I'll start the class again. So I just want to warn you, if that happens, it's not you, it's my house, something here. Oh, so many things going on. Okay, so let's talk about lab. So lab of this week, we will be having lab this week. And here's why, or here's the backstory. I'm hoping you can see that. Can you see my, yes. So one of the things that I am world famous for is predicting ice conditions, particularly when the ice is going to leave a lake. We are going to talk about this in more detail after spring break when we come to the chapter on heat, temperature, and ice circulation. But it's very important to us right now because once the ice is rotten or weak, we will not go out on the ice. But the lake will still be mostly ice covered, so we can't do anything else until the ice goes out. Once the ice has gone out, we can collect water from the shore. If somebody has a canoe or a kayak, some people could go out on the water and collect water samples. But it's pretty important for us to know ahead of time when these events are going to occur. So here's how you predict when the ice is going to be rotten and when the ice is going to go out. It correlates very well to a cumulative warmth model. And what I've got here on this spreadsheet is, this is Excel, which I do lots in, right? A high temperature each day, the low temperature each day, and then what the average temperature was for that day. So what you see on this, and this is this year. Oh, it says 2020. It's actually, yeah, this is actually 2021. And so that's what this chart shows you here. The high temperature each day is in blue. The low temperature each day is in orange on my screen anyways. And then the gray is the average temperature for each day. Right? And here's that really cold stretch. Remember that? Like when this is Celsius. So, even the high temperatures were below 10 below. And we are here in the beginning of March. I think actually we're right here on this cold day stretch here. And then the temperatures are going to be again. After today, these temperatures are all the forecast temperatures. So these are the real temperatures so far this year, and then the forecast temperatures on the right. Now the way this model works is any day that the average temperature is above freezing. So here the average temperature was 1 Celsius, and that is this day back here. It just adds 1 to this running total. So this column here is a running total. Now here it was 2, it's actually 1.5. Anyways, it rounds to 2, and so then it goes up another degree. And then it doesn't change again until the daily average temperature for the day is above freezing. Okay, here we go, February 22nd, the average temperature for the day was 1. So the running total goes up by 1, it goes from 2 to 3. Next day, the average daily temperature was 3, so this running total goes up to 6. The next day, average temperature was 1, goes up to 7. Next day, it was below 0 for the average temperature, below freezing. And so the temperature, the running total did not change. So this is a running total of the average daily temperatures when the average daily temperature is above 0 Celsius. This is called a degree day model because this is a sum of degrees times days. And this is only counting days when it's warm enough to melt. So this is a melting degree day model, abbreviated MDD, melting degree days. Okay. So this is a melting degree day model, and I'll show you this again after spring break. But for now, let me simplify this a little and show you this. Okay, so that's the melting degree day total, rising whenever the daily temperature is above freezing. And when the daily temperature is below freezing, it just stays the same. So it's melting degree days. It's not, no freezing in here, only melting. And here's where we are right now. Or actually Sunday when I made this model. All right, what if we add in the forecast data? Well, oh, here's the point. When the melting degree day total passes 80 melting degree days, the ice on Lake Phelan will be rotten. Just based on the last several years of observation, it takes about 80 melting degree days to make Lake Phelan rotten. When the melting degree day total passes about 110, the ice will go out as soon as it is windy. It is so rotten that the ice will go out as soon as it's windy. And in fact, on average, it's about 110. So what we're doing with this model is predicting when will the melting degree day total hit 80. On that date, that's our prediction, our forecast for when the ice will be rotten. And then when will the melting degree day pass 110? That's our forecast for when the ice will go out. Okay, so what I'm going to do now is I'm going to put in the forecast data. That was observed. Now we're going to add the forecast data. And here is the forecast. This was as of Sunday. And you know, the farther out in the future you go, the less stable the temperature, forecast temperature is. But as of Sunday, the ice was forecast, based on the weather forecasts as of last Sunday, the model says that the ice will be rotten around March 30th, and the ice will go out around April, 6th or so. And you could look at the chart to see when 80, you could look at the table, right? And here's 80, somewhere between March 29th and 30th. Here's 110 in between March 5th and 6th. Okay, so I'll be updating this model because, of course, the actual daily temperature tends to be different than what was forecast 2 weeks ago for the date. So I'll be updating this, but basically what this says is that we have 2 more labs on the ice, right? We're going to have this week, no problem. if we had class next week, we could go out on the ice next week, but it's spring break. And we will probably have good ice the week after spring break as well. But after that, based on current weather forecasts, the ice will be rotten. We won't be able to go out anymore. Okay, so this is what I utilized. This is what I used in order to refine the lab schedule for the next couple of weeks. Do you have any questions about this? Oh, okay. All right, so let me end this. Yeah, we could say that. Screen sharing has stopped. So we're gonna have lab this week, and I'm pretty confident that we will have lab the week after spring break. So what are we gonna do with this time? Well, this week, we are gonna go to Lake Phelan, to our usual place. We are going to do lab 6 officially. Actually, we started lab 6 a while ago. I just never announced it. And lab 6 is lab skills. It's a lab skill lab. You're gonna learn how to do a bunch of things, use a compass, use a GPS unit, tie 2 knots, coil a rope. I think there's something else. Lab 6 is lab skills, Limnology field skills, and it's posted on D2L. We are also going to collect some data that we will use for labs 7 and 8. So on Thursday of this week, we're going to meet at Lake Phelan. We will be doing lab 6 in preparation for the lab test on lab 6. But we're going to officially learn how to triangulate using a map and compass. And while we're doing that, we're going to collect the lab, some water samples and lab data for lab 7 and lab 8. Lab 6 will be assessed not with a lab report, but with a lab practical test. And that will be after spring break. Probably, possibly, depending on the timing of things, possibly that first Thursday after spring break. If I have too many things planned for that one day, we will do it the following week, or however that works. Anyways, so there's no test this week, but we will be officially doing lab 6. Okay, so lab 6 involves lab skills. One of them is using the GPS. Oh, right here. Using the GPS to find your location and describe your location. Using the compass and tying some knots and coiling and securing a rope. Now, you know there are 2 kinds of limnologists, those who are annoyed and those who are annoying. There are limnologists who coil ropes neatly and tie them. And there are limnology students who don't. And I'm not even saying they're going to become limnologists, because it's kind of hard to keep your job when you annoy your boss, right? So I would much rather you learn how to tie, that's correct, how to tie knots and coil your ropes neatly. Okay, so now I'm going to demonstrate. What order do I have these in? I'm going to demonstrate these knots. Oh, actually, I'm going to pause and say, hey, if you have a piece of rope handy, that would be really handy. A piece of rope or a piece of string. I'll demonstrate these now, and then we'll do it again after the break. Okay, so we got 3 kinds of knots that we want to learn. Square knot is first. So let me see if I can get Jeffrey to turn his camera on. Hey, Jeff, turn your camera on. Okay, so I got, this is Jeffrey over here. All right. There's so much going on my little tabletop here. Here we go. Okay, I got some rope. And I'm just going to take the end of this piece of rope here. All right, how many people know how to tie a square knot? I think this is an important life skill. Useful in all kinds of situations. James says yes. Good. So this week, lab 6, we're not going to work in lab groups particularly. Well, we have a finite amount of equipment and you will need to team up. And I would like you to try to team up with somebody who doesn't know what they're doing, or if that describes you, team up with someone who does. So people who know how to use a compass, I'd like you to try to team up with people who don't, just so that you can help them learn. So everybody learns. Okay, so let's tie a square knot. I'm going to show this to you, right? It's pretty simple. Here's one rope. Here's the other rope. Okay, so how does it go? Right over left. Let's find it. Here we go. Right over left, and then you tuck it under, so it looks like that. And then left over right, and then tuck it under, and then that's your knot, and pull it tight. Now, if you've done your square knot correctly, it will look like this. Where's my camera? Like this, right? And the beauty of a square knot, you see it's kind of like two loops grabbing each other. Symmetrical, like that, two loops kind of grabbing each other. It's quick to tie and it's easy to untie because all you do is let one side go slack and then you can just pull on, push it and then pull that down and then you can untie it. So a square knot is a great knot for tying two ends of ropes together when the two ends are about the same, the two pieces of rope are about the same diameter. And it's a kind of a temporary, it's a great temporary knot. And it's like tying your shoes, right, without the bow, right? So it goes right over left, left over right, makes a knot that's tidy and tight. Oh, did I say that right? Left over right, right over left, right over left, left over right, makes a knot that's tidy and tight. But it's also easily undone. So it's not a great knot for, making a permanent connection between something that's going to be like jiggling around or back, if there's any kind of flexing of the rope back and forth, like tightening, loosening, tightening, loosening, tightening, a square knot will come undone. So it's great for something that's under continuous tension, but not for something that's going to flex back and forth like this. If it's the only knot that you know, and you want it to be more secure, you can do this. Take the free end, and make make a little half hitch there. Take the other free end, wrap it around, make a little half hitch there. And that can turn a square knot into something more permanent, also harder to untie, and that's optional. On the lab test, I will give you a piece of string, a piece of rope. Every person will have a piece of rope, and I will ask you to make a square knot. And then if you do it right, you get a point. And if you do it wrong, you don't. This seems kind of simple, doesn't it? So that's a square knot. And it's universally known as a square knot. Okay. Any questions about the square knot? You can Google it. There's lots of demonstrations on the web for square knots. Okay, that's a square knot. Now, what if instead of tying two ropes together, you want to tie something onto the rope? Now, it could be like a fish hook. Or it could be some like weird piece of lab equipment, like one of these thingies. Or, I got a lot of, limnologists, we have a lot of things tied to ropes. Well, this is not a square knot. This is a bowline. It's spelled bowline, and it is pronounced bowline. And a bowline is a knot that you use to tie a thing to a piece of rope. You use it to make a loop at the end of a piece of rope, like you would tie an object to it, or you would tie a rope to something. But if you need a loop at the end of a rope, you will use a bowline. Here's how you do it. Let's see if I can demonstrate this. So there's my device. All right. This is a little trickier. And let me walk you through it. First of all, you've got your rope. One end is the short end. This is the bite. And this is the long end, the one that leads like all the way over here. You take the long end, the long piece, and you make a loop in it. And with this, you got to make sure, with this loop, the part that goes to your object is got to be on top. So here, this is on top of that one. So you're making a little loop, and you got to have this part on top. This is on top. Then you take your loose end and you pull it up through the hole that you've made, up through there. Go around down behind this and go back into the hole. Then you could grab the two pieces that go through the hole, grab those two, and pull it tight. Now you've got a nice loop at the end of the rope, in this case attached to a piece of limnology equipment. Very secure, but easily undone. It's kind of the same kind of knot that if you flop one end down, you can kind of pull on one of the loops, just bend the loop, and the whole thing can be undone easily. So it is a very secure knot, and yet easily untied. It's kind of the two magic things, right, for a knot. It's secure, it doesn't come undone when you don't want it to, but it is easily untied. Okay, so let's do that one more time. Okay, so we're going to make a loop in the rope with the upper strand being the one away from you. And then, this is the bunny rabbit, right? The bunny comes out of the hole. Bunny goes back around behind the tree. And bunny goes back into the hole. And there's your knot. Okay, so I will, Google around and look to see if I can find a good video on the web, because I think there are many. And it's a very standard knot spelled B-O-W-L-I-N-E. So if you just Google bowline, how to tie a bowline, you will get better visual imagery and explanation of how to do it. And on the lab test, I will give you a piece of rope and I will give you a washer or something, and I will say, attach this to your rope with a bowline. And then you tie it and write your name on it, throw it in the bucket, and if you did it right, you get a point. If you did it wrong, you don't. So now, just as I said with the square knot, if you tie a bowline and you're really kind of anxious to be sure that it's doesn't come undone, and in limnology, sometimes we have really expensive equipment tied to ropes and we don't want it to come undone. You can take the free end and just make a couple of half hitches and that makes it really secure. But that's just kind of bonus. Another thing you would do is if you wanted to do this permanently, I would lay these together and wrap some duct tape around it so that this can't come loose. And when I tie a knot that I want to be sure my students don't untie, I will take the free end and the main stem and wrap a piece of duct tape around it, because I think nobody in their right mind undoes a piece of duct tape, you know, because it's annoying for one thing, and it's clearly not meant to be done and undone, done and undone. Okay, so that's a bowline. Any questions? I use the bunny comes out of the tree, bunny comes out of the hole, goes around the tree, goes back in the hole mnemonic. But I have had students use a different mnemonic to do the bowline, and there is and they make perfectly good knots. So there is a different mnemonic also out there that you can use, and that's fine. Questions? All right, you know, I will attempt to demonstrate these outdoors in the class. Yeah, we'll be practicing this in this lab, but I also thought that it would be kind of, it's kind of hard to show, you know, what my fingers are doing when we're outdoors and everybody is 6 feet apart from each other. So I figured I would demonstrate this during class, instruct you to go look online to find better videos of it, and I will look for those too. So yeah, when I say we are doing lab 6, what I mean is we're going to practice all of the things that are on the lab 6 assignment. Okay, so that's the square knot, and that's the bowline with the tip of the hat to the half hitch, which can be used for extra security. Now let's talk about how you put the ropes away when you're done. Now, in some of the cases, what we've got in lab is some kind of a spool to coil the rope or the string on, but not always. Big spools, small spools, polyester spools, whatever. One thing you'll notice is that when you spool and unspool rope, sometimes you get these like twisty bits here. Like look at how twisted that is. And there's 2 reasons for this. In this case, the reason is if you wrap the rope on a spool like this, but then you unspool it like this, you actually introduce twist into the rope. And it's actually fairly common for some reason that it's easier to spool it one way and then unspool it the other way. So it's pretty common for rope to end up with twists in it. Now, if your object is hanging freely on the end of your rope, then as you pick it up, it will naturally just spin, you know, releasing the twist in the rope. But that isn't always true. But sometimes it's helpful to stretch the rope out so that when you coil it up, I can't see that. When you coil it up, the twist is released. Now this rope is braided, but some rope is just strands twisted together. And some rope has a natural coil to it. So that when you spool it, you have to pay attention to which way it coils. So now let's talk about how to coil a rope. We don't have spools for everything. You will not have spools for everything in your life. Sometimes you will have a coil of rope and you need to secure it. There are 2 ways to do that. This is one way. Here's the coil and here's the tie. The other way is something like this. Here's the coil of rope. So it's kind of messy rope, but it's secured in the middle like that. Because what we do with our rope is we put it in a box with other stuff. it's better to have the second type. This is a version of a gasket coil. So let me show you how to do it. First of all, in limnology, you always want your coils to be 1 meter long. So it's good to know how long a meter is. So let's just pretend that this is a meter. You want all of your loops to be the same length. Once you've coiled it, and you've got about a whole length left, or two, you put the coils together. I'm going to make these smaller just to make it more visible here. Hold on. Okay, you got a coil. They're all about the same length. You got about twice this length left over. This is what you do. Here's your nice coil. Oh, let's go back to Jeffrey's camera here. Okay, so here's my coil. There's 2 ways to do this, 3 ways to do this. I'm going to show you the easy way. Okay, so here's my piece at the end, right? Here's my rope. I'm going to take my piece and coil it around the outside of the rope back the way it came. So like here it is going around. I'm going to coil it back the way I came. So this way. Okay, so now I've got a nice, neat, tidy, wrapped all the way around. Here's where it started. Here's where it finished. So you take where you finished, go back to where you started, put it under there. Now you're basically making a square knot. I'm upside down here. A square knot. Uh-oh. So left over right, and then right over left, or even if you only do it once, and then right over left, and then, oh, got a crappy knot there. Okay, and now you've got it secured. So you're basically tying a square knot at the very end, or at least starting a square knot at the end. The other thing to do, which is the correct way to do it, which I will tell you even though I find it harder, is you go back, you've got your coils like this. You take a loop of here, hold it on like this, shove it through there, and then pull the whole coil of rope through this little loop that you made. And then pull it down tight, snug it down. That is called a gasket coil or a buntline coil knot. It is the correct way to do it. I never do it this way, but I think I probably should. And I hope that some of you will someday get jobs as limnologists. You know, Minneapolis Parks is always looking for summer interns in their Parks and Rec limnology department. Anyways, you should practice doing this, a buntline coil, this type of knot where you make a loop at the end and pull it through. I have posted on D2L a nice demonstration of somebody doing that. I asked my brother how to do it and if he could send me a video, and he dug up this video of a guy who kind of looks like my brother, actually. So you could go look at that. The most important thing is that I want you to be able to coil the rope neatly, hopefully in 1 meter long loops, wrap the rope around it multiple times, and then you take the free end of the wrapping and tie it back to the beginning, and then that makes this coil kind of like one big knot that holds it together. Now you can take this and throw it in the equipment box, and it's not going to get all tangled up with stuff. So this is better than this type of attachment for our purposes. This is very similar. I think you can see just where the knot is. Okay, that's what I want to say about knots. I urge you to get a piece of string or whatever when we take a break at break time and come back with a shoelace or something so you can practice. And we will practice this with rope on Thursday. Hopefully some of you already know how to do this or you pick it up quick so that we'll be able to team you up with somebody else who maybe needs a little bit of help practicing. Because, yeah. All right, are there any questions about the knots and coiling part of today's content? Questions, comments? How many people can already tie a square knot? Can you give me a little check if you can do? Got a couple of, how many people you're like, no, I really don't know how to do a square knot. Oh my God. Yeah. Can I have a mix? Okay, good. And then, how about this? How many people have ever done something like this before? Coiled a rope and tied it. Anybody? I got one yes, one no, two no's, three no's, two yeses, four. So we got kind of like a 2-to-1, no to rest. Okay. We'll work on this very important skill. In any field of science, especially at the entry level, handling the equipment correctly is just so critical. I mean, what do you learn in chemistry class? You learn how to handle equipment. How do you handle a micropipetter and not break it? How do you, I don't know what else they do in chemistry. How do you use a funnel? I don't know, whatever. A hot plate without burning yourself. You get a job with a bachelor's of science degree and, if you're lucky, you're going to be working in somebody's lab. And what are they going to care about? Are you going to follow the instructions? Do you handle the lab equipment correctly, even without them telling you how to handle it correctly? They're like, she's got a college degree. She's a biology major. Of course she knows how to do this. I didn't think I had to tell her. Oh my God. What a disaster. Yeah, you don't want to be that person. You want to be the person who just automatically, whatever, cleans the microscope stage correctly when you're done. Uses only lens paper on the optics of a microscope. Never leaves a micropipetter lying down with stuff in it. Always hangs it vertically. Those things. Just, I can't even tell you how important it is. And it's true in limnology too. We're not a well-funded field. You hire an intern and they break your equipment. It's like, great. You don't want that. You want someone who takes care of stuff. Okay. Next, triangulation. Yeah. Okay, so this is what you're going to do. You got a compass. And you want to find a place on a map. The lake map. Now, one of the really distinctive things about lakes is that there's really not a lot of landmarks that you can see. You could be here, you could be there, you could be somewhere else. How are you going to find a particular place on a lake? If it was a forest or field or whatever, there'd be rocks and trees and, hills and valleys, but on a lake, it's like flat and it's all water. Furthermore, a lot of lakes are really big. So there's two ways to locate specific places on lakes. One of them is with GPS coordinates. We've talked about what those UTM coordinates are, Universal Transverse Mercator, the UTM coordinates. And you know that the GPS works by listening to satellites. So here we are on the surface of the Earth. Here we are on the surface of the Earth. And you are, somewhere, here's you. distance you. How does your GPS unit know where you are? Well, up here in space, there are satellites. And the satellites are all broadcasting signals, in all directions. And that signal includes the identity of the satellite and the time. And it is known where the satellites are. So the GPS unit has a clock in it and a receiver. And when it receives the signal from this satellite, based on how long it took the signal to get from the satellite to the GPS, it knows the distance. The satellites are so far away that the signal takes a little bit of time to reach you. And because the signal includes the time at which the signal was sent, then that was very loud. Then the GPS unit knows how far away it is from this satellite. Similarly, this satellite is sending out a signal with the time and its identity, and the GPS unit can tell how far away it is from that satellite. And hopefully there's more than 2 satellites that it can hear signals from, and there may be even a suite of satellites. And by knowing the distance to each of these different satellites, the GPS unit can calculate where on the surface of the Earth you are and your elevation also above the Earth, so that your 3-dimensional location. This is not triangulation. There's no angles involved. It's all about distance. So this method is called trilateration. Now, it's called trilateration because it takes at least 3 satellites to be able to get your 3-dimensional position. Typically, it's going to be listening for more than 3 satellites. 5, 6, 7 even would be ideal. But it's called trilaterization, and the latter refers to distance, like lateral, right? So the GPS does not determine your location by triangulation. It does it by trilaterization, by knowing distances to 3 or more objects. Do you have any questions about how a GPS unit works? So GPS stands for Global Positioning System. And it was originally developed by the United States military, I believe, and has gradually become more and more improved, and also more and more available just for ordinary consumer citizen use. So GPS stands for Global Positioning System, I think. Global Positioning, what else would the S stand for? Yes. Okay, any questions about GPS? So the GPS unit in your hand is not a transmitter. You can't use it like a radio or anything, or a phone. It just has a receiver. And it listens for the signals which are carried on radio waves of some kind. I don't know what wavelength. All right, so we got our knots, we got a rope coil and secure, GPS, trilaterization. Okay, so now let's talk about using a compass to find a location. Of course, GPS has not been around forever, whereas a compass has been around for a much longer time. And here's how it works. Let's look over here to Jeffrey, see if I can demonstrate on a piece of paper here. So let's say that you want to describe a location on a lake. This is Lake Hiawatha. I just had some extras of this piece of paper. And you have some landmarks that you can see. Here's a fixed landmark that's really quite visible. I'm gonna make it super visible by painting it red. And let's say that, now here's a benchmark, and that's a really well-known thing, but it's not going to be visible. Here's the outlet to the lake, right there. And maybe there's some kind of structure at the outlet to the lake that you could see. You could look over there. Oh, there it is. So if you wanted to tell somebody how to get to this point here, let's say you've looked for the deepest part of the lake, and you're standing, you know, right here in the deepest part of the lake. What you could say to them would be, you draw some lines on one piece of paper here. The streets right here are north, south, east, west, so I'm going to use them just to make my lines here. And it's a vertical. Get some nice vertical lines there. Okay, so from this point here, we want to describe the direction to this landmark. This is north. This is north. So we're going to describe this angle here. So this is Hiawatha, so this is the recreation building, you know, and there's a fishing dock right off the end here. So you can take a compass. Jeez. They promised not to drop the tree on the house. Like they promised that like 3 or 4 times, which started to make me a little concerned. Yeah. Okay, so we got a compass, and the way compass directions work are north is up, south is down, east is to the right, west is to the left. If you don't know which way is east and west, practice, because this is really a key piece of information. If you've drawn a north-south line and you're looking at it, east is to the right, west is to the left. Similarly, the numbers go around clockwise. North is 0 degrees, east is 90, south is 180, west is 270. So you stand on the point, you take your compass, and you look at your landmark, and you turn the dial on the compass so that you can get a very precise direction. Now with the sighting compasses, there's a mirror, so you can look down on it at the same time as you look forward, which enables you to be very accurate. You turn the dial until the red needle is in the red shed, and then the direction that the compass is pointing has got the letter and the number right on the dial lined up. So you stand here and you sight at your landmark and you go, oh, that is 45 degrees from where I am standing. From me to that landmark is 45 degrees. And then you look at your other landmark and you describe the other angle. So from me to this landmark is, you know, I don't know, like 140 degrees. So in other words, this landmark is northeast of me, at an angle of exactly 45, a compass direction, an azimuth, if you want to use a technical term, the compass direction of 45 degrees. This landmark is southeast of me at a compass direction of 140. And you can write that down. And then somebody else can find this spot by walking around with their compass, right? Until they are at a 45-degree angle from that landmark and a 140-degree angle from that landmark. You know, and there's ways to do this efficiently, right? You stand on one of the landmarks and you head in the correct direction. And then you just keep walking in the correct direction with your other eye on the other landmark. And then when you get, so you start here and you'd walk along, you go, okay, I gotta go that way. So you head that direction with one eye looking to the right, looking at your compass until you are at the right angle to that one, the correct angle to that one. Because this involves two angles in 2 dimensions, and if you wanted to get a elevation as well, you would need a third angle. It is called triangulation. And with 3 landmarks and a good compass, you can triangulate your location, you know, quite precisely. So what we're going to do on Thursday is we're going to go to Lake Phelan. We're going to practice tying knots and coiling ropes and triangulation. We're going to go out into Round Lake and make some measurements there and collect some samples. And then we're going to go to Phelan and do the same thing. And then we will be all ready for lab 6, 7, and 8. Do you have any questions for what we're going to do this week or any of this labby instructions? Do you want to see a picture of my oak tree? Here, look at this guy. You see that guy up there in the tree? See that little guy up there? Yeah, there's my oak tree, my fence. There's the guy in the tree that is cutting it down right now. It's a very sad day. Huh. All right, any other questions about lab? Like, anybody scared? Is anybody like, oh my God, it's above freezing, the lake's gonna melt? Are we all good? You know, I didn't invent triangulation. That's probably not the best explanation you're ever going to hear of triangulation. I'm sure if you like Google it, you could come up with, you know, I think there's a lot of people out there who've made little videos of how to triangulate things. And the same thing goes for the knots. I kind of gave you my version, but I'm not going to claim that, you know, I'm the best version, the best explanation of that there is. All right. Well, our next topic for today is a pure lecture topic. We have worked our way down the food web. We went from fish to zooplankton. We took a digression over to plants, which are very important in creating structure in the food web. Even if they're not really part of it, they really help structure it. And now we're going to get back onto our food web and talk about algae. Algae are photosynthetic, and they are the base of the aquatic food chain. Okay, so what I'm going to do now is show you some PowerPoints, and then we're going to take a halftime break. If anybody wants to practice some knotting, you can do that too. I'm going to turn Jeffrey's video off there. All right, let's see if I got my PowerPoint fired up here? Okay, so we're going to talk about algae. Just before class today, I saved this onto the D2L site. So if you want, you know, your own copy of this PowerPoint, it's on D2L right now. All right, let's see. When we talk about algae, there are a couple of different ways to categorize them, to divide the algae into categories. I mean, there's tens of thousands of species of algae. I don't know how many. There's a lot. And you can't just be thinking about every single one of them individually. There's gotta be some way to group them into meaningful groups so you can just talk about the groups of algae. So one of the ways that we can categorize algae is by habitat. So let's start by thinking about the algae if we categorize them by habitat. Okay. First of all, you know, when I say algae, I mean the photosynthetic green stuff that is not plants. So this is the base of the aquatic food chain. So we can categorize them in various ways. The first way we're going to talk about is by habitat. Now, when we talk about the algae in lakes, the two major categories are the planktonic algae and the attached algae. So we can talk about the planktonic algae or phytoplankton. And the word plankton means wanderer, wandering in ancient Greek. So these are algae that, you know, they're basically drifting in the water, carried by the movement of the water. Now, it's true, some of them have flagella and they can swim a bit. Some of them can control their buoyancy so they can float more up to the surface or sink more down. But, you know, compared to something like a fish or a muskrat, you know, they really cannot move. They're more moved by the water than they are moving through the water. So some people say a synonym for planktonic is free floating. You know, kind of passive, more carried by the water than deliberately locating themselves. Now, within the planktonic algae, we can talk about things that spend their entire life cycle in the water. And that would be things that are holoplanktonic. They spend their whole life cycle as plankton. But there are other plankton that only spend part of their life cycle free floating. They spend part of their life cycle as plankton, but part of their life cycle on the mud as benthic algae or on plants as epiphytic algae. And those that spend only part of their life cycle are called meroplankton or meroplanktonic. Spell it planktonic. Meroplanktonic or meroplankton. Plankton, yes. And then if you were to go out and take a water sample and look at the algae in it, you would find some algae in your water sample that are not normally really found out in the open water. They don't need to be, it's not part of it. They're simply there because they're dislodged from their normal habitat, stirred up from the benthic algae or washed off some of the plants where they were growing. They're really only out in your plankton sample by accident or by luck. And I use those words because that group is called the tychoplankton. And tycho is a root word that means luck or chance. I don't know of any other words in English that use the root word tycho. I guess I could look that up pretty quickly, but I don't. And so if you were to go out and catch a plankton sample from a lake and look at the algae in it, most of the things that you would find would be holoplankton, but depending on the time of year, there might be quite a few things that are meroplankton. And there's always a few random things in a plankton sample, a few random things that aren't really planktonic algae at all, but easily dislodged from where they normally live. And those are the tychoplankton. Now, the other extreme from the planktonic algae would be the attached algae. And some people, attached algae is kind of a long word. So it's also common to call these epiphytic algae. Or actually, I'm going to use epiphytic in a moment, or the periphyton. Now, periphyton technically means growing on a plant, on top of a plant or something. So it doesn't really apply to all of the different versions of attached algae, but it's a common word and people recognize it when you say it, periphyton. Oh yeah, it's part of the periphyton. They're like, okay, that's the algae that grows attached to stuff. Now the true periphyton would be epiphytic. And epiphytic means growing on top of plants. P-H-Y-T, phyte, on top of plants. That's the epiphyton. But there is also attached algae that lives attached to rocks. And that would be epilithic. So epilithic is on rocks. Epiphytic is on plants. There are some types of algae that you find specifically, typically growing on mud. And that would be epipellic. And I think the root word pellic refers to clay, very, very fine particles. Now, mud tends to be very nutrient rich. And if you live on the mud surface, you can expect there to be a lot of nutrients available in the pore water of the mud just underneath you. So compared to almost anywhere else in the water, right on top of the mud is a high nutrient location. So there are algae that you'll typically find just forming a film, a biofilm on top of the mud, if there's enough light reaching the bottom. And of course, the third definition of the littoral zone is the depth in which there is enough light reaching the bottom that benthic algae can live on the bottom. And these would be primarily epipellic algae. So anywhere in the lake where enough light reaches the bottom that algae can live on the mud, that's the third definition of the littoral zone. The first one is, enough light reaches the bottom for rooted plants to live. Rooted plants live there, it's littoral zone. The second one is 15 feet or less. The DNR uses that because it's quick, right? You can just look at a lake map and do it 15 feet or less. And the third definition is enough light reaches the bottom that algae can live on the mud. And that would be like basically the compensation depth, right? So we've used all three definitions. Okay, so in terms of periphyton or attached algae, we have algae that live on plants, that's the epiphytic algae. Algae that live on rocks, that's epilithic. Or algae that live on mud, that's epipellic. Algae that live on sand are episamic. And this root word PSAM refers to sand. And you could see, I think, that living on sand would be very, if you were microscopic algae, it's very different living on sand than living on mud. Mud is full of nutrients, very fine grain. You can live on top of it. Maybe you're just going to get worried about getting covered with silt. Episamic, that's sand. Sand does not hold nutrients. Sandy soil is nutrient poor soil. But the thing about sand is if you're a little kind of algae and you're on the sand, you could like sneak down into the sand when it was dangerous and scoot back up again when it was safe, and sneak down in between the sand grains and then sneak back up again. And there are diatoms that do that. They go down into the sand a little bit and come back up and go down and come back up. You can't do that in mud because mud is very fine grained. There's no like spaces really. And there tends to be no oxygen below the thin skin on the top of mud. But in sand, there may be oxygen fairly deeper in. So different kinds of algae are going to live on mud than live on sand. And the last category, which I hear a lot about at meetings recently because it's just really kind of interest to people, is epizootic algae. And can anyone guess where do epizootic algae live?
On animals?
On animals, that's right. So, you know, manatees have algae that live on them, special, maybe not super special, but there are distinct species of algae that live on manatees and are epizootic algae. Whales, of course, are famous for their epizootic algae. And I was just reading about, it was in the newspaper this morning. What was it? Some other thick-skinned animal that has its own algae that lives on top of it. Okay, and I think that I have mentioned with respect to mud and sand that mud is finer grained and sand is bigger particles. Sand, silt, and clay, right? Sand is the biggest particles. Mud is silt and clay. And when you're someplace where the waves are crashing on the shore and there's much more water movement, the finer particles tend to get carried away constantly. So often very close to shore, it'll be sandy, and farther away from shore, it'll be muddier. And that's just typical. In fact, you can reconstruct the depth of oceans based on the grain size in the sedimentary rocks that formed from the sediment. That's an aside. Okay, so one of the ways that we can classify the algae, categorize the algae, is based on their habitat. Another way that we can classify the algae is by their size. If you have any questions about any of this before I erase it, I just want to give you quickly some size categories. All right, and I mentioned, you know, that mud is going to have more nutrients and sand is going to be nutrient poor. So we can also categorize algae by size. Which you'll remember we did with the zooplankton. Now it makes sense to categorize zooplankton into net size and smaller size, things that are big enough you catch them in the net, things that are too small, things you can kind of see with the naked eye, things you must use a microscope to see, just for practical human purposes. But in terms of the lake itself, the size of the zooplankton is important because what they eat is determined by how big they are. Generally speaking, zooplankton can only eat things significantly smaller than themselves, right? So a great big zooplankton is not going to eat another great big zooplankton. Great big zooplankton could eat a little one, no problem. Sweep it into their little mouth. What about the algae? Size in itself is important for algae too. The size of the algae is going to determine how rapidly it sinks through the water. It's going to influence who can eat it. And so in some cases, a small algae, it might be beneficial to be small just because it's small. Small surface area to volume, slow sinking rate. Other times it might be beneficial to be a big alga. Big alga sinks more rapidly. Big alga has poor surface area to volume ratio, for nutrient uptake by diffusion. But a bigger alga is not going to fit into the mouth of a copepod or a cladocera or something, certainly not like a little rotifer. So if there's a lot of herbivory, maybe, being bigger is good. So size in and of itself is important to the algae. So these are the size classes. The smallest of all is going to be the femto. And these are the things that are under 2 microns in size. That's tiny. That's not even really phytoplankton. It's really viruses and bacteria. And small bacteria. Okay, so that's not algae, but it is stuff that you would see if you were collecting algae samples. Then you get picoplankton. These are things that are sort of 0 to 2 to 2. I didn't do that right. There's a decimal place there. Two-tenths of a micron. So from 2/10 to 2 microns. These are things, this is now big enough to see with a light microscope. This would be cyanobacteria, which are true algae, and small bacteria, or just regular sized bacteria. The next one is going to be nano, because of course these names are just taken from the, milli, micro, nano, pico, femto, the metric prefixes. So the nanoplankton are like from 2 to 30 microns. And this includes a lot of things. This includes most unicellular algae. Cryptophytes, euglenas, chrysophytes, green algae. Most unicellular algae is in this category, 2 to 30. You can see this on 100x, 40x microscope. If we were collecting algae samples and taking them to the lab, we would see lots of these little things. But microplankton is easier to see because it's really bigger. It's more like 30 to 70 microns. Now you'll remember 70 microns is pretty close to the mesh size of the zooplankton nets we use. So this is algae that's big, relatively speaking, like you could see it easily with a light microscope, but you're likely to miss most of it if you're using a net to collect the plankton. So just from human practical terms, you can see these types of algae very easily with a light microscope. And most algae is in this category, but you're going to miss it if you're using a plankton net to collect it. Now this includes most of the rest of the algae, most multicellular algae. And what do I want to say this? What I really want to say is most big diatoms fit in this category. And the sort of small colonial algae. So you got stuff like, I don't know, chrysophytes. We haven't seen any of these algae yet, so I guess it doesn't do any good to use their names. I'll use some names and I'll tell you how big they are. So we're going to look at some pictures, and there will be among them small colonial algae too. I'm thinking Volvox, is kind of pushing the upper limit of these things. And then when you get to the bigger algae, you get above micro. Well, we got sort of two categories of micro, small micro and big micro. And the reason we're going to use this bigger category here is because this is the net plankton, right? Above 70 to 200 microns. That's really pretty big. That's 0.2 of a millimeter. So this is like the size of the zooplankton in the jars. Like you can see things that are 0.2 of a millimeter. You can see things that are this big, this big. And what is this? This includes things like most big colonial algae. And then you get up to stuff that isn't even really plankton anymore. You get beyond that, and it's really not, I don't know, this is really big. So 0.2 to 2 millimeters. And now this is very big, right? Great big sheets and filaments of green algae. So when we were collecting macrophytes, and some of what you pulled up was not plant at all, it was filamentous green algae. That's in this category of mesoplankton, like really too big to be even planktonic at that point. It's just draped on the plants, right? Hanging on stuff. So for algae, that's very big. And it's things that are sheets, things that are long threads. Filaments is the technical term. Okay, so you can classify the algae in terms of where it lives. Is it, broadly speaking, planktonic or attached? We can talk about the algae in terms of its size. Is it small unicellular stuff? Is it great big colonial things? Or is it somewhere in between? Is it big enough to catch in a net? Or are you mostly going to miss it if you use a net to do your sampling with? Is it, if it's really small, then it's going to get eaten by really small zooplankton, like rotifers and ciliates. The really small stuff is going to get eaten by the small zooplankton. The sort of medium-sized stuff can get eaten by your typical voracious herbivores like the Daphnia. The really big stuff can't get eaten by the Daphnia. It's too big. So in terms of the place that things fit in the food web, for algae, a lot of it depends simply on how big they are. If they're unicellular, they're going to occupy one place in the food web and get eaten by one group of herbivores. If they're medium size, they're still going to be photosynthetic, but they're going to get eaten by different herbivores. And if they're really big, they might not get eaten at all. Great big clumps of blue-green algae, of cyanobacteria, nobody can eat them. So the size alone is an important aspect for diatoms, true, yes, for diatoms, but also for algae in general. Okay, so we talked about classifying the algae by habitat, classifying the algae by size. Now let's talk about classifying the algae by taxonomy. So I'm gonna start PowerPoint to show you a little bit of the taxonomy, then we'll take a break, and then we'll come back and look at some more taxonomic groups. And I'll tell you right now, there's 7. 7 is a magic number. Okay. Oops. All right, I'm going to use the word algae in the broad sense of the word. Sensu lato is Latin for in the broad sense. And what I mean by this is I use the word algae to refer to any little green things in a lake that are photosynthetic, except plants, not plants. There are other people who say the cyanobacteria can't be called algae. They exclude the cyanobacteria because they're not eukaryotic. That would be a more narrow sense of the term algae. We'll use sensu lato again. It's a Latin word, taxonomists use it, and it just means in the broad sense, as opposed to sensu stricto, which is in the strict sense, in the narrow sense. And you use it when a word has more than one definition, and one of the definitions includes the other one, plus stuff. Okay, so let's start with a picture from Calf. He shows you some pictures of some really cool algae. And we're going to have a lot of great labs this semester outdoors, but we're not going to use microscopes. So we're not going to see these things up close. And trust me, they're green. They're not black and white, they're green. So here's a bunch of different algae. They have a lot of different shapes to them. Here's a beautiful colony of Asterianella, a diatom. Here's a great big scary Gymnodemia, Serratium, which is a dinoflagellate. Got a couple of kinds of blue-green algae that form these big messy blobs of stuff. Pediastrum, or just a beautiful bright green algae. Some algae have flagella, as you can see. Some algae have cell walls with silica or scales made of silica, like glass. Some algae make blobs around themselves of gelatinous goo. That'd be the blue-green algae. Some of them have hard cellulose armor on the outside. That's a dinoflagellate here. Some of them are unicellular. Some form colonies. There's a huge diversity of forms among the algae. When we talk about their taxonomy, we typically group them into 7 groups. Let's see if I can get my 7 groups right here. Now, there's some dispute about what is a kingdom and what is a phylum and so on. The kingdom Protista, which was a very useful kingdom, it included everything that was eukaryotic and wasn't plant, animal, or fungi. Well, Protista's really pretty much been abandoned because it's not monophyletic. But it was very useful because it was an easy way to describe these things. So all of these are eukaryotic except cyanobacteria. Our 6 major groups of eukaryotic algae are the euglenas, the diatoms, the chrysophytes, the cryptophytes, the dinoflagellates, and the green algae. The green algae are the most closely related to plants. And some people say that they actually belong in the plant kingdom because the plant kingdom is not monophyletic without them. Yeah, anyway. And then the cyanobacteria are important, abundant, photosynthetic little green things in water. But they're not eukaryotic, so some people say you can't call them algae. So we're either going to call them cyanobacteria or we're going to call them blue-greens. And sometimes I'll say blue-greens, blue-green algae. So these are the 7 groups: euglenas, diatoms, chrysophytes, cryptophytes, dinoflagellates, green algae, and cyanobacteria. Let's talk about the cyanobacteria first. They are the only ones that are prokaryotic. Okay, there's also the purple sulfur bacteria and the green sulfur bacteria, which we will talk about later, but not today, because they're not a major group. They occur in very strange places. Okay, so we got the cyanobacteria that we're going to call blue-greens or blue-green algae. They're prokaryotic. And what that means is they don't undergo sexual reproduction. They can divide really rapidly because all they have to do is like duplicate everything inside and then separate. And that's really fast in terms of cell division and sort of reproduction. Generally speaking, cyanobacteria cells are very small. When you look at them under a microscope, if you see a small cell that's green and you can't even see the chloroplasts, it's so small, it probably doesn't have chloroplasts. It would be a cyanobacteria. Only eukaryotic organisms have chloroplasts. They do have chlorophyll. They do photosynthesis, and they do have structure inside the cells. They just don't have separate membrane-bound organelles inside the cell. So when you look at them with a microscope, they're green, but it's kind of just grainy green, like dots of green all over, not chloroplasts. They're all unicellular. Some of them are colonial. Did I say that wrong? They are often unicellular, and they can be colonial, but they're never truly multicellular. Filamentous blue-green algae that are important that we will talk about are Anabaena, Oscillatoria, and Aphanizomenon. There are also cyanobacteria that make kind of blobby colonies like this. Like, I don't know, is that a colony or is that just like a bunch of cells stuck together? I don't know. It's a blob, and that's microcystis. Very common in Twin Cities lakes. Very common in South and Central Minnesota lakes. And of course, you may have heard of microcystis. Anybody know why have you heard of microcystis? Anybody ever heard of microcystis?
Is it toxic?
Microcystis, some strains of microcystis produce a chemical, microcystine, that is toxic. So when people talk about toxic algae in Minnesota, they're usually talking about microcystis. Now, and not microcystis don't always make this chemical, but when there is toxic algae problem, it is microcystis that's causing it in Minnesota. The cyanobacteria have a bacterial cell wall, and they also make some protein that they put on the outside of their cell wall, which is called a gelatinous sheath. So, and you can see that in kind of in this sketchy diagram. The dotted line outside is showing sort of clear goo inside. Here's one here, and I'm not sure if you can see, but these cells are all stuck together just by something kind of clear. Now, gelatin is a protein, long fibrous protein that traps water and forms a gel. And mixed in with the gel tends to be other chemicals as well that make the cyanobacteria taste bad, make it hard for them to digest, organisms to digest it, or can even be toxic. Yeah, and when there is a taste and odor problem with a drinking water supply, it is often because of the cyanobacteria and chemicals that are in the gelatinous sheaths. The cell wall is bacterial, as I've mentioned, so it's got peptidoglycans in the cell wall, which other eukaryotic organisms don't have. They do not have flagella, but they can control their buoyancy with structures known as gas vacuoles. This is not a true vacuole like a plant vacuole. These are actually very curious cages of proteins inside the cell. When they do a lot of photosynthesis, they can trap oxygen inside the structure as a bubble, which lowers the density of the cell as a whole by expanding its volume without increasing its weight much. And that makes the cell as a whole lower density than the water, so it can float upward. This enables them to control their buoyancy, which gives many cyanobacteria the ability to control how deep in the water they are floating. That's an important characteristic that cyanobacteria have. And I think I said it backwards. I think I said oxygen. I meant carbon dioxide. So when they release CO2, they can fill a vacuole with CO2, which enables them to float up where there's more light. If there's more light, then they'll be doing more photosynthesis and consuming the CO2. If they do too much photosynthesis, they might exhaust all the CO2. This will make them more dense, and then they can sink down a little bit. It's not good to be too high in the water if you're algae, because if you're right on the surface, you can get bleached by the sun and actually damaged. So algae really like to be a little bit below the surface. And in fact, for any kind of algae, there's like some perfect light level. And being able to control your buoyancy so you could like go up to your perfect light level or go down to your perfect level or back up again, you know, as the sun comes and goes, is really a major skill, a very beneficial talent that the cyanobacteria have. Some cyanobacteria, like the Anabaena, which is maybe cartooned here, actually do make a couple different types of cells, the most important of which are called heterocysts. And in the heterocysts, cyanobacteria can fix nitrogen, meaning they can take nitrogen gas that's dissolved in the water and convert it into something useful, like amine groups. If you can convert nitrogen gas into, you know, ammonium, then you can use it. But ordinarily, you can't use nitrogen gas for anything. So if there's a nitrogen shortage, the algae that can fix nitrogen have a definite advantage. And the only algae that can fix nitrogen are cyanobacteria. And even so, not all of them, only the ones that can make heterocysts. And there's a reason for that, and that is the enzyme that does nitrogen fixation, nitrogenase, binds irreversibly to oxygen. So if you have a cell that's doing photosynthesis and releasing oxygen, and it's trying to do nitrogen fixation, it's going to fail because the oxygen released by the photosynthesis will bind to the nitrogenase, and then it can't do any nitrogen fixation. Well, then Anabaena and the other nitrogen fixers in this group have different cells that are actually clear. They have no chlorophyll in them. And all they do is nitrogen fixation. And then they release the nitrogen through portals to the other cells, and they take sugars in. So that's a special talent that only the cyanobacteria have. And a few organisms that have cyanobacteria as endosymbionts. Yeah. So if you can't fix nitrogen yourself, why not take in some cyanobacteria and keep them in your skin and let them fix it for you? There's an idea. Well, I wouldn't do it personally, but lichens do it. Some diatoms do it. Coral coral animals do it. They make coral reefs. Some of them do it. So, and the other thing they can make is resting cells, thick wall. It's just like a regular cell with a really thick wall and real slow metabolism. And it can fall into the mud and just rest there for years, even until it gets stirred up again into the sunlight and goes back into active life. That's, I think, what I want to say about cyanobacteria and what I want to say before we take a break. So it's a little bit past our traditional halftime break, so let's take a 10-minute break. Go get a piece of string or a piece of rope if you can find one. Shoelaces work well, or just go get a cup of coffee, and I will see you in 10 minutes. All right, welcome back. Anybody get a piece of rope and work on your knots? Do this with a little thing here. Make a little tie. Make your square knots. And you'll know it's a square knot if it looks like two loops like this. Then you'll know you did it right. Pretty simple. There's really only one way to do it wrong. And if you do it wrong, it's not right. I think square knot refers to the fact that it's symmetrical, right? Both, it's not really a square, but it is symmetrical both ways. So you look at it this way, you look at it that way. Maybe it's not symmetrical. I don't know why the name is. Maybe it's called square knot because it's the right knot. You know, like a right angle is, yeah. Anybody have any questions about lab? Oh. Anybody? You're all good. Shall we go back to algae? All right, let's go back to algae. I know I'm talking a lot today, but you know, do you want to see this guy? Yes. You see that little guy up there? Where is he? Where'd he go? There he is. Look at him. Oh, look at that. He's cutting this damn tree down. The neighbors are coming by with their little kids. I can't believe how many of the little kids are in the neighborhood. They're all here holding somebody's hand, looking up at this guy in the tree. Yeah. Oh my God. I know. It's really sad. We bought this house because of the tree. Yeah. I love that tree. I love that tree. But Baroque blight. Yeah. It's really terrible. What are you going to do? Yeah. Okay, algae. Algae. Let's talk about algae. Yeah, I like algae too. All right. There are two groups of algae that are really incredibly wonderful. And one of them is diatoms. And the other one is diatoms. And then in third place is cyanobacteria, because they have a lot of really cool characteristics. The blue-green algae, they have skills that no other algae have. You know, being able to control their buoyancy, no one else can do that. Making toxic chemicals that poison humans and dogs, okay, almost nobody else does that. Some of the dinoflagellates do. What else can they? Fixing nitrogen. Almost nothing on Earth can fix nitrogen. Really, it's just cyanobacteria and some fairly closely related soil bacteria. And that's really it for fixing nitrogen. Am I forgetting anything? Almost everything else I can think of off the top of my head. The only reason they can fix nitrogen is because of a symbiotic association, either with cyanobacteria or with the soil, the rhizobium and Brady rhizobium. So cyanobacteria are really cool, and they have an enormous kind of economic importance. And they have this medical health connection by making this liver toxin. And why do they make that, you know? Yeah. Anyways, but moving on. Okay, it's going very slowly. Why is that? Okay. Let me, I gotta pull Jeffrey down here so I can just make sure that you see what I think you see. Okay, so let's move on. Now we're gonna talk about eukaryotic things. First of all, let's talk about the euglenas. Euglenas are really kind of cool. They're kind of creepy too, because they're unicellular. They got a flagella. I mean, they have an eye spot. so they can swim towards the light or away from the light, so they can swim and move themselves through the water to maintain optimum light levels by virtue of having a flagella and an eye spot that senses light. They can't see things like images, but they can sense light intensity. And the eye spot is right next to the flagella. So that makes sense. Here's a microscope view of a euglena, and you can see the flagella here caught in motion as it's like coiled up in a crazy coil right next to the cell there. One of the things that I said they were kind of creepy, and that's because they're photosynthetic, but they can also absorb organic matter from the water and burn that for energy, like respire it. They have mitochondria, because they're eukaryotic. But so they can be both autotrophic, making their own sugar with their chloroplasts, but they can also absorb organic molecules from the water and send those to the mitochondria and use that to make ATP. So they're both autotrophic and heterotrophic, which means they are mixotrophic. And some members of the euglena group don't have chloroplasts at all, in which case they function as zooplankton, effectively. Yeah, so they would be heterotrophic nanoflagellates. If they didn't have a chloroplast, they would be heterotrophic. And they're tiny and they have a flagella, so they're heterotrophic nanoflagellates. Okay, right now we're talking about them because they often have chloroplasts and are algae, function as algae. They are not very closely related to plants, but when they do make too much sugar, they turn it into starch. And one of the cool things and unique things about them is inside their cell membrane, they have a structure that's like bands that wrap around the inside of the cell membrane that are made of protein and are kind of flexible. They're sort of like flex steel. And that's what enables them to maintain a shape and to kind of change their shape. They can kind of get bigger and spread out and get bigger and spread out because of this sort of protein banding inside the cell membrane. And that is called the proteinaceous pellicle. And I think that the word pellicle is related to pelt, like animal pelt, I think. But in any case, they're the only ones that have this. And euglena is the classic example of a euglenoid, as the group is called, or their technical name, euglenozoa. Because they are studied historically by zoologists, the name of the group is euglenozoa or euglenophyte, euglenophyta, depending on who studies them. If the zoologists study them, the phylum is called Euglenozoa. If botanists are studying them, they're called euglenophytes, Euglenophyta. Yeah, so they're really mixotrophic. They're really on that border between plant and animal. Yeah. So that's the euglenas. Euglenas make nice pets because if you forget to feed them, they die and then they just eat each other. Yeah, so I don't know if that makes them a good pet or just creepy. Yeah, anyways, euglena. They make a good laboratory animal for that reason. Is they're as a population, they're pretty robust, meaning, easy to care for. Because if you neglect them or the power goes out, they have this backdoor food supply. Yeah. Okay, enough of that. Let's talk about these. Anybody know what these are? Look at all these different cells here with all these different shapes. Who knows what these are?
Diatoms?
Diatoms. Oh, how did you know that, Abdul?
I think I've seen pictures of these before.
Yeah. Well, I show it in every class, right?
Yeah, that's probably where I saw it then.
And this is a diatom picture from, I don't know, Wikipedia or something, so it's pretty easy to find on the web. And diatoms are unique. Each of these is a single cell organism. It's got chloroplasts every one of these is photosynthetic, and every one of these has a cell wall that is made of a lot of silica, silicon dioxide, in other words, glass. And this enables them to hold beautiful geometric shapes rigidly and perfectly. Like once it's made, that's its shape. And they make, you know, different species can be identified by the shape of their cell wall and the patterns on the cell wall. So these are diatoms. These are mostly marine, possibly even all marine diatoms. These triangular ones, we don't get those in fresh water. What do you need to know about the diatoms? Okay, every diatom is a single cell organism. Sometimes they stick together after they divide, forming distinctive colonies. Here from the first picture of kelp is a Asterionella. Asterionella makes asterisk-shaped colonies. 1, 2, 3, 4, 5, 6 cells. Originally it was one, maybe this one. It divided, each of those divided and then divided. So they reproduce by cell division mostly. Here is an Alocasyra or possibly Melosyra. Some of them, most of the freshwater Melosyras are now called Alocasyra. And this is one cell here. If you can see, this is one cell here. And the way that the, let me outline it here. Here's one cell. And you're looking at it from the side, and diatoms in side view look like that. They are two halves. I'm looking around for something to demonstrate this with. Another good example. A diatom cell is kind of shaped like this. You can see this. It's got two valves, and the cell would be inside. So the diatom might be shaped like this. And then connecting the two valves is like a couple thin, very thin, flexible bands of silica. When it's time for the diatom to divide, what it does is this. Let's say this was our original cell. The cell content inside the cell divides and it pushes the valves apart from each other. And then and then two new valves form in between. And so if I can make so the bottom one becomes this and the top one becomes this. Let me see, go back. So we had one original one, which is the two blue ones. They divided and then they formed two new valves inside. Now you've got two cells. This is the original valve of one. Here's the original valve of the other. The two valves in between are the new valves. So now you've got a chain with two cells in it. Then if each of these divided, you'd have a chain with 4 cells in it. This mode of cell division of diatoms is distinct and unique to them and creates some of the interesting situations that diatom populations experience. And I've done a bunch of modeling on this. So there's a poster in the hallway of the Science Education Center, maybe more than one if you want to learn more about diatom cell division and its consequences for the distribution, the distribution of sizes within a population. Obviously, over time, oh, I didn't say that. The key piece is the two new valves that are made inside the old ones are each a little bit smaller than the parent valve it inherited from the parent. And so the two offspring cells, one is going to be the same size as the parent and one will be a little smaller. And over time, the average size in the population gets smaller and smaller and smaller. And the range of sizes in the population gets bigger and bigger and bigger as the minimum size gets smaller and smaller. Yeah, cool stuff. Anyways, The cell wall is called a frustule. Frustule is made of two valves. The valves are a protein superstructure which silica precipitates onto from the inside of the cell. Cool biochemistry. This makes the cell rigid and kind of heavy. So they sink faster than other algae do, other phytoplankton. So that you might think this is a problem, really, because they're going to sink faster. And if they sink out of the photic zone, they're dead. They can't support themselves anymore, right? With photosynthesis. But some people say it's an advantage because if you sink faster, you're constantly moving through nutrient-rich water. As a cell, you're absorbing nutrients from right outside. And this causes a little nutrient depletion zone on the outside of any cell. If you move through the water, then you're moving into water with more nutrients in it. So people say that might be beneficial to sink rapidly. I don't think so, but anyways, could be true. The thing that I think is beneficial about this cell wall structure is the cell wall structure is really incredibly fast to make. The diatom accumulates silicic acid inside, extracted from the water, and then when it's time to divide, they make a framework organized by microtubules. The microtubules organize, they make a scaffolding, and then they release the silicic acid into a vacuole containing the scaffolding, and just like chitin fibers. And then the silica just precipitates. And they can make a cell wall in hours, which is really fast, with very little energy expenditure. They accumulate the silica inside, they make chitin fibrils into the pattern, and then they just open the valve and it precipitates, meaning, crystallizes in a sense. Very, very energy efficient way to make a rigid cell wall. Alternatively, you could make it out of cellulose, like plants do and green algae do, but cellulose is a long chain of glucose molecules, right? You're wasting sugar. You're using sugar to make a cell wall, and it takes energy to make make all of those, do all that dehydration synthesis, constructing the cellulose. So making a cell wall of cellulose uses a lot of sucrose and takes a lot of energy, glucose, and takes a lot of energy. So this is a very efficient way to make a cell wall, and look at all the amazing shapes that you can make with it. It's hard to make a unicellular thing, anything that's not a sphere. I mean, a sphere is the natural shape for things. It's the smallest surface area to volume. It equalizes all pressure. If you want to be unicellular and be something other than a sphere, you have to be rigid in some way. You have to find some way to push and maintain a different shape. So silica really enables a wide variety of shapes, which can have a variety of benefits. Like an Asterianella, even though it's very big, sinks slowly because it's like a fluff of a dandelion. It functions like a parachute because of its shape. So anyway, diatoms are really cool. That's what I want to say. Okay, what about their life cycle? The cells, generally speaking, reproduce by division. Generally speaking, they don't have flagella. The only time they have flagella is during sexual reproduction. One of the gametes has a flagella. Sexual reproduction is pretty rare in the diatoms. Like in some species every year, in a lot of planktonic species, decades. Most of their reproduction is cell division, just vegetative cell division. Some diatoms have a little raphe on them, which is a slit through which they can exude some kind of mucilage like snail slime, and that enables them to scoot along on surfaces. So diatoms can dive into the sand and climb back up, scooting around the surfaces of the sand grains. They're phototrophic. Some of them can fix nitrogen because of symbiotic nitrogen fixers. Some of them can absorb organic molecules from the environment, which makes them heterotrophic, so therefore mixotrophic. When they do too much photosynthesis and have extra energy, they store it as, not starch, chrysolaminarin, which is a unique lipid, unique to the diatoms. They reproduce by cell division, which decreases the average size of the population, and rarely by sexual reproduction. And here are the names of some diatoms you're going to hear again. You've already heard me say Asterianella. You've heard me say Elycosyra and Melissyra. I haven't used any of these other names. If we were looking at diatoms and microscopes from Round and Phelan, you would see all of these species. This Cyclotella, Staphynotiscus, Fragillaria, Tabellaria, Synedra. Okay, well, I got lots to say about diatoms, but I think I want to stop there with talking about diatoms at least. And clear my drawings and move to next group. Oh, pictures. These are what diatoms look like. These are diatoms from, any lake in the Twin Cities. This looks like an Asterianella, one single cell. Here's a long skinny Synedra. Here's some naviculoids. They might be like scooting around, scooting around on the slide. Here's a Tabellaria, which in side view and face view looks like this, but in side view looks like a square box. Closely related to the diatoms are the Chrysophyta, the chrysophytes. We know that they're closely related because they're the only other group of algae that does anything with silica. Some of the chrysophytes make little scales that go on the outside of their cell. Each one of these little bristles has a little disc that is on the surface of the cell. So they're called scales, like fish scales. Yeah. Some chrysophytes make little glass vases that they live in. This is Dinobrion or Dinobrion. Here, Dinobrion or Dinobrion. And here's a microscope image. And here's the cell inside with the flagella inside this glass lorica. When they do too much photosynthesis and have extra energy to store, they store it as chrysolaminarin. They, generally speaking, reproduce just by cell division, and these colonies are formed by the cell dividing and the next one getting added on at the end, similar to how diatoms divide. Rarely, but not never, by sexual reproduction. So you can see they have a lot of things in common with the diatoms. I have less to say about the next couple of groups, the cryptophytes, the cryptomonads. I don't know what to say about them. I don't know that I've ever knowingly, I'm exaggerating, knowingly seen a cryptomonad. They're so cryptic. I mean, really, what can you tell about it? It's unicellular. It has flagella. It's got chlorophyll. It's phototropic. And if you saw that, you would be like, okay, it's a little green alga of some kind with flagella. Could be anything, I mean, it could even be a diatom gamete, it could be a green algae, could be a euglena. You'd look at it, you go, okay, it's not a euglena. The shape's not right. It does have kind of a distinctive shape and kind of a distinctive color, but they're really not very distinctive. And they're always unicellular with two flagella. They are fairly common, but I think frequently misidentified or overlooked. The most common species is Cryptomonas, which means cryptic cell. Another common one is Rhodomonas, which you might guess, what color do you think Rhodomonas is? Like based on its name, what color do you think it is? red is the correct answer. That's right. So this root word H-R-O-D-O, rhodo, means red. Like Rhodomonas, no, that's this one. Rhode Island Reds, Rhodesia. Not really sure Rhodesia is named. Yeah, anyway, so that root word road. Okay, that's the Cryptomonas. That's all I got to say on them. Dinoflagellates are cool, but they're too scary for me to really like them. First of all, they're bigger than other types of algae. So you're looking along a microscope slide, it's your little algae, and all of a sudden, bam, there's this like giant thing, and it looks like it's like a space invader. Or, you know, it's like one of those things that suddenly you look up and it's hovering over the surface of the earth. And, you know, yeah, that's what these look like when you're looking at a microscope. They're unicellular. They're called dinoflagellates. And dino meaning spinning, and flagella meaning flagella. They have two flagella, one in the front that pulls it forward, and one in a groove around the middle that makes it spin. So one flagella stays in this groove and kind of flips back and forth, thrashes around in the groove, and that causes it to spin as it moves. Now, some people say the reason that they're called dinoflagellates is the dino, like dinosaur, like terrible monster, right? Because dinoflagellates do a bunch of biochemically like scary things. First of all, they cause bioluminescence. Waves are crashing in the ocean. It's dark and the stars are overhead and the waves are glowing. Yeah. You walk along the sand as the water has washed out and is draining out towards you. Walk on the beach and your footsteps glow blue, you know, like Good King Wenceslas, right? You know, like walking in the, yeah, whatever, Christmas carols. Yeah, so they cause bioluminescence, and it can be kind of scary or really cool, like spiritual cool, you know? They also cause red tides. When too much organic matter washes in the ocean, in an estuary like Chesapeake Bay or the Gulf of Mexico, and you get an algal bloom there. Algal blooms in the ocean are typically not blue-green algae. In lakes, it's cyanobacteria, typically. But in the ocean, it's typically not. It would be more likely to be dinoflagellates. And dinoflagellates, some of them make a neurotoxin, which accumulates in shellfish. So shellfish, like mussels and clams, filter the water and eat the zooplankton that they catch. And if the algae and stuff that they're filtering in and eating contains this chemical, that chemical just accumulates in the shellfish. And then when people eat the shellfish, they get paralyzed, or they get, you know, shakes, or they get, you know, burning sensations in their hands. Yeah, and that's what red tides do. And dinoflagellates are responsible. I don't know why they're called red tides. My understanding is they're not particularly red in color, but it may be more red like danger, more like a symbolically red tide. I don't know. Some dinoflagellates shed this cellulose armor, and the squishy little cell inside lives symbiotically in coral, the animals that make coral reefs, the coral animals. So that's cool. And that's what I want to say, I believe, about dinoflagellates. There are some species of dinoflagellate that live in fresh water. This is one of them, and you do occasionally see it. There are some lakes in which dinoflagellates, instead of cyanobacteria, dominate a summer algae bloom. I'm not really sure why. Why would it be dinoflagellates instead of cyanobacteria? I think that pH may be an issue. Cyanobacteria don't do very well in acidic water. So if the pH is too low, then it might tip the scale in favor of these. If you've got a dense algal bloom in the summer, the only algae that are going to really thrive are the ones that can get above the other algae, right? A dense algal bloom creates a lot of shade. If you're an algae that, like a diatom that's falling constantly, you're always going to be in the shade of the other algae. So who's going to win when there's a ton of algae in the summer? Diatoms, phytoplankton that can float, like cyanobacteria, or something like dinoflagellates that are a hefty swimmer. Yeah. Now let's go to the group that is edible, the most easily edible, most closely related to plants. Has lots of nice things to say about it. Incredible diversity of forms. In the green algae, there are unicellular algae, there are colonial algae, filaments, spheres, sheets like lettuce. There's, it's incredible diversity of form in the green algae. Some examples are Chlamydomonas, which you may have heard of. It's unicellular, it has a flagella, it's a common lab rat kind of algae. For people who study, whatever, cell recognition, there are different strains of Chlamydomonas, and they will only reproduce with a different strain. So it's kind of, interesting. Okay, unicellular without flagella, chlorella. You can buy powdered chlorella at the health food store if you want to juice up your smoothie. There are some that make plates, like these two. Here's Cenodesmus, here's Pediastrum. This is kind of like a flat plate of cells with curious shapes. Cenodesmus is always a set of 4 with 2 spines on the ends. Pediastrum forms these kind of disc-like colonies. Each of these is an individual cell. 1 cell, 2, 3, 4, 5, 6, 7. So kind of cool. Of course, everybody loves Volvox. And Volvox is a spherical colony. The individual cells have 2 flagella, and they're arranged all in this big open ball of a sphere. Some green algae form filaments, one of the most famous of which is Spirogyra. So here's an individual cell of the Spirogyra, and here's the chloroplast. And the chloroplast is a ribbon that winds through the inside. It's not like a little disc like plant chloroplasts, it's a ribbon. Green algae have all kinds of different shapes of chloroplasts, and that's kind of a cool thing. When they have flagella, they have 2, and they are at the front. They pull it forward. They make cell walls out of cellulose, just like plants. When they have extra energy, they store it as starch, just like plants. They do photosynthesis using chlorophyll A, chlorophyll B, and they have carotenoids, the same pigments as plants. They're photosynthetic. They reproduce by cell division, but also by sexual reproduction. And there are 3 major classes within the Chlorophyta. The Ovophyceae, which is the sea lettuce class, Charophyceae, which is related to Chara, which is a macrophyte that was in the book, the aquatic plants book, and Chlorophyceae, the green algae. So lots of, not a lot of biochemical diversity, but lots of morphological diversity in this group. Now these are not in the big 7, but also important in some curious microhabitats are the purple sulfur and green sulfur bacteria. They're prokaryotic. They do photosynthesis, but not normal photosynthesis. Instead of splitting water to get the electrons and releasing oxygen, they split hydrogen sulfide and release sulfur as the byproduct. So instead of, in normal photosynthesis, water is split and it's the source of electrons and protons and oxygen. In the sulfur photosynthesis, hydrogen sulfide is split, and that is the source of the electrons. And sulfur is released as a byproduct. The only place that you find the purple sulfur and green sulfur bacteria are places where there's no oxygen. Why is that? Because they need hydrogen sulfide as their electron source. And if you have oxygen and hydrogen sulfide, you get sulfate. It destroys the hydrogen sulfide. So you can only find these where it's anaerobic and there's hydrogen sulfide and light, enough light to power photosynthesis. That's a really unusual situation because typically if you're up where the light is, there's going to be oxygen. So the two places where you find them are on the surface of mud that's anaerobic because of decomposition in the mud, but there isn't water movement down. So you can find them growing like in marshes where the plants prevent the water from moving. So the water is anchored in place in a sense. And under the surface of the water, it becomes anaerobic. And so the surface of the mud where there's sulfur is anaerobic, so therefore hydrogen sulfide. But because the water's not very deep and there's no other algae there, there's light for photosynthesis. Situations like that. The other place you find them is at the chemocline of meromictic lakes. Meromictic lake is a lake that doesn't mix all the way to the bottom. Therefore, the bottom water never gets refreshed with oxygen. And it becomes a highly anaerobic layer of water at the bottom of the lake. So you can have the whole bottom of the lake have hydrogen sulfide dissolved in the water. At the top of the anaerobic bottom layer of water, there might be enough light for photosynthesis, and you would get a layer of purple sulfur bacteria. We're going to look for purple sulfur bacteria on a lake that I hope might be meromictic. And that's what we're going to do in lab 8. Lab 8 is meromixis. Yeah, we'll see how that goes. Or maybe it's lab 7. Lab 8. Yeah, 7. Anyways, after spring break, we're going to hunt for meromictic lake. And one of the things we're going to do is look for a layer of purple sulfur bacteria. And we'll take water samplers, we'll put them in the water, pull up water from different depths. You can put the camera down and you can see the layer. A meromictic lake that I work on, sometimes brownie, there'll be like a layer of like pink water, like 5 meters down. Lake's really, really deep, like 50 feet deep. Go down a certain depth. And then there it is, a layer of pink water. And it's the purple sulfur bacteria just hanging at the top, at the chemocline, at the top of the chemical layer. Very cool. There's a little picture. That, anyways, unicellular, phototrophic, obligate anaerobes. They can only live in an anaerobic environment. There's other weird things about them. They have one of them has flagella, one of them has gas vacuoles, but we can say more about that later. Okay, now I have a question for you. You ready for this? We have 7 groups of algae. There were taxonomic groups, but now we're going to rearrange those groups and lump them together into what limnologists call the 7 functional groups. So this is like our 4th way to categorize them, and this is kind of by function. First of all is the nitrogen-fixing blue-greens. Like, I don't care what species they are, what genus they are. If it's a nitrogen-fixing blue-green, it goes in this group. Then you got the other blue-greens that don't fix nitrogen. Then you got the diatoms. Then you got colonial green algae, dinoflagellates, and everything else. The last group, it says 7, but there's 6, aren't there? Did I forget one? I should change that to 6. The other group is the pigmented flagellates. Unicellular, have a flagella, have chloroplasts. That's all it takes to be in this group. It doesn't matter what your taxonomy is. If you're unicellular, you have chloroplasts, and you have a flagella, you're in this group, pigmented flagellates. And they tend to kind of swim together, so to speak. This includes cryptophytes, which are always unicellular with flagella. Euglenas, they're always unicellular with flagella. Some of the chrysophytes, some of the green algae, Some of the green algae are unicellular with flagella, and they go in this group. Now, this doesn't include all possible algae, but this covers most of them. And these are the groups that you need to think about when we think about algal ecology. So when we think about the ecology of algae after spring break, we'll use these groups. Okay, now we've got 7 or maybe 6 groups of algae, 7 taxonomic groups or 6 functional groups. I have 3 questions for you. Now I'm assuming that you paid attention to this lecture, which I don't know whether you could have or not. It's kind of boring to just listen to somebody talk. But you know, hopefully you've been paying attention or reading the textbook while I've been talking and you have this information. So let's say that you have this information about these groups of algae. I have 3 questions for you, and I'm going to put you in breakout groups and ask you to try to answer them. These questions are in the PowerPoint, which is posted on D2L, but I'm just going to read them for you in case you don't have access to that. The first question is, which types of algae will you get in a plankton net, and which ones will you miss? So if you're somebody who studies algae and you collect them with a net, you're going to see some groups more than others. Which ones are you going to see? Which ones are you going to miss? And why? The second question is about ecology. Let's say that we've got a lake, it's got a lot of algae in it, but it's also got a lot of herbivorous zooplankton or zoobenthos. Lots of little algae-eating animals. What, all other things being equal, which types of algae, which taxonomic groups and which functional groups are you going to find in such a lake? And why? And the third question is, here we go. The third question is, let's say that you have a lake that is eutrophic. It has lots of phosphorus because there's lots of humans in the watershed, and we just put phosphorus into water. We do. We put it on our lawns. We release it in our sewage. You got humans, you're going to have phosphorus enrichment of your lake water. What type of algae are you going to find in a eutrophic lake, in a phosphorus-rich eutrophic lake, and why? And the opposite question, which is kind of a different question, is what type of algae do you find in low nutrient water, in oligotrophic water, water that's very clear, and why? This might be 2 different questions. Okay, so I got 3 questions for you here. Hopefully at least one person in each group has all 3 questions. I'm gonna let Zoom put you into some kind of random-ish breakout rooms and invite you to solve all 3 of these questions. Just start with number 1, and then I'll stop at some point and have you report out. And when you're done with 1, move on to 2. But does that make sense? Okay, so let me create some groups here. Let me just make sure nobody's depending on Jeffrey, because he doesn't pull his own weight. All right, the rooms are open. Take these questions with you. Come back with some answers.
Should we make these on a whiteboard?
If you want.
Or I don't know. Do you have anything to write down?
I don't. I definitely was paying attention, but I don't know. I don't know any of the answers. I'm so I was listening and I don't know. I'm afraid of this. Afraid of these questions. Do you know any of these answers?
Mm, I feel like I know some that could go under the what we would catch in a net, but that's about it.
Yeah.
Otherwise, I have no idea. I felt like none of the what we went over is related to these questions.
I know. Yeah, I agree. Part of me is like, let's just go to Google and see what we find, but I don't know. I wonder if she's gonna pull the whole this is from the textbook thing. I don't know.
Probably.
I'm gonna grab my textbook real quick. Maybe it'll be there. I don't know.
Did you find anything in the book?
No.
Nothing.
I flipped through a lot of pages, but I don't think I mean, I'm looking a lot at like the macrophytes section, and it's either macrophytes or else it's like, it's either plants or else it's zooplankton, but there's not a lot about algae in the book.
That's what I thought because I went through the book a little bit yesterday.
Yeah.
I think my guess would just be the green algae.
The green algae?
Reason why. Oh, because let's see. I think looking at the size, at least on the PowerPoint,
Yeah,
well, the diatoms are really small,
they are.
and I think the other ones, I don't know about gold brown, but like the cryptomonads are like 5 micrometers, so those are small. I don't know how big the dinoflagellates are. Those are unicellular. I think usually when you're classifying on size, it's in most unicellular would be like 2 to 30 micrometers, which is not in the net.
Right.
So I don't know.
That one makes the most sense. The green algae.
Just because I think it's colonial also, which would I feel like it'd be more of a mass,
Right.
and then you could catch it. Because the only other colonial one I think is the, what is that, golden brown?
I think so.
Yeah.
I just feel like none of them are gonna be the crypto ones, the cryptophyta,
Yeah, I don't think so.
because she was not enthusiastic about those at all. She was like, I don't even know what these are.
Well,
I
yeah,
never
she's never
see
seen them.
Yeah. So I think
I
we're
think
trying to rule that out.
is the cyanobacteria too small?
That's a good question. Because aren't there some cyanobacteria? Because that's blue-green algae.
Yeah.
I would feel like that one would be big enough too. the golden brown algae, the green algae, and then the blue-green algae. I feel like all three of those would work.
That's what I would think. That'd be my best guess, anyways.
Yeah. At this point,
I'll take a
I'm
guess.
guessing. Yep. And did she say kingdom specifically?
It
Or was
says
it?
kingdoms or phylum,
Yeah.
so either or.
And then I guess the ones that we would tend to miss are the diatoms. I don't know.
Yeah.
Sure. Okay. Well, that one's done. um, number 2, which will dominate where grazers are abundant?
I think it's just the like cyanobacteria. Did you read any of the textbook?
No.
So wait, do you have the PowerPoint pulled up?
Yeah.
Look at the next slide and you see the 2 diagrams.
Yeah.
I think so. If I remember correctly, I think controls that talks about like the grazers, and that's where they're present.
Okay.
And I think exclosures is when they're not present. Might be a different type of grazer though. Can't remember which. Oh yeah, 24. So it's in chapter 24.
Yeah. I definitely saw those graphs. Yeah, it says it says note the seasonally increasing importance of large blue-green algal epiphytes in nature controls while in the absence of macrobenthic grazers exclosures.
Okay.
So the diatoms remain dominant for much of the season, only to be replaced by green algae. So green algae would take over in the yeah. I sort of had something. But green algae takes over in the controls part. Which
Okay.
is what you were saying.
Yeah, is that when they're present? When the grazers are present?
Yeah. Those are when the
Okay.
grazers are present, and then when they're not present, the diatoms take over.
Okay.
Huh. Okay. So which will dominate when they are abundant? That one is green algae.
Would it be blue and green? Or just green?
Oh, sorry, blue-green algae.
Okay. I didn't know because cyanobacteria is a blue-green, right?
Yeah.
And then it also has greens on the diagram.
Yeah. Okay. And then it's talking about phosphorus-rich waters and not phosphorus-rich waters.
This I
I
have no clue.
24.10, the title of that section is eutrophic or eutrophication and benthic plants.
Oh really?
Mm-hmm.
Oh wait. I'm just looking at the next diagram in the PowerPoint. That one's based off phosphorus. So is that the cyanobacteria again?
Yeah. From the are you doing it from the PowerPoint?
Yeah.
Yeah, and then, yeah, that would be the cyano. bacteria. Because that one's high in phosphorus, and then the low would be the dinoflagella. Right?
Yeah, I think so.
Okay. All right. Well, we'll be somewhat prepared when she asks our group a question.
Give it our best go.
Yeah.
Back, everybody. So we had some breakout rooms here. I think we had like 4 rooms. So maybe we'll have some different answers here. Why don't we start with room 1? Did you guys, that'd be Jang, Kat, and Sam T. Did you guys get an answer to this first question?
What we discussed was, or what we came up with was the algae, so like the golden brown cyanobacteria, which is blue-green, and then just the chlorophyta,
Mm-hmm.
green algae.
And
That we said is you can catch in a plankton net.
okay, and why would you catch that why would you catch those in a plankton net?
I think some of those might be larger, but also I was thinking it might be because of the colonial aspect. So it comes in more of the mass, easier to catch possibly.
Yeah. So I got a cartoon of a plankton net here, and you used a net, and you pull it through the water, and the water goes in here as you pull it upward and comes out through the mesh, and the silk has openings that are like little squares. And a zooplankton net typically has a mesh size somewhere between like 70 and 100. I think the zooplankton nets that we use have a mesh size of about 80. So generally speaking, if an alga is bigger than that mesh size, it's probably going to get caught and stay inside the net. Whereas if it's smaller, it will tend to go through the mesh. Now, there'll be some exceptions, it'll get caught on the edge or something, but generally speaking, if it's smaller in diameter than the mesh size, then you're going to miss a lot of them. So that's the principle. And you're saying that the ones you're going to catch, I thought that was a good list. You said colonial blue-greens, like the filamentous ones or the ones that make big blobby things like microcystis is a big blobby thing, or Anabena, that's the chain. They'll get caught inside the net. Big colonial green algae, like Volvox. Volvox is really big, and it's not going to make it through that hole. It'll get stuck. A filamentous green algae, like the ones you were pulling up last week, with the weed hook, those are going to get caught inside the net. Really big diatoms and colonial diatoms. And I think you mentioned colonial chrysophytes, like Dinobrian or Dinobrian, however you say that, chrysophytes. So these are all things that you're going to catch in a plankton net. But everything else, you're going to miss, especially pigmented flagellates. Right? Those like unicellular things with flagella, you're going to miss those. They're too small. So, yeah. So I think a good thing to bear in mind is that in terms of like data that you would see from lakes, you know, if the algae has been collected with a zooplankton net and the zooplankton net has a mesh of 70 or 80 microns, then any sort of phytoplankton data is going to be really missing, well, especially the unicellular ones, chlorella, chlamydomonas, cryptomonads, they'll be missing. All right. Any questions about that? Did I get your answer, Sam? Did I do that up there okay for you?
Yep. Yeah.
Yeah. All right, let's go to our next question. Oh, okay. All right, so let's say we've got a lake and there's a lot of Daphnia, a lot of rotifers and a lot of Daphnia. What kind of algae are you going to find in that lake? Anybody have a good answer to that? How about James, Keenan, and Sam, the other Sam? Did you guys get an answer to this?
For this one, I just kind of winged it. I was just for zoo benzos. I just kind of assumed that maybe it would be the algae that had flagella because they could escape potentially and swim away. That was just kind of my for at least the zoo benzos that are kind of bottom feeders in the sediment. That was kind of my logic and thinking.
Yeah, you were just winging it. I'm hearing
Yeah.
that. Yeah, I'm hearing that. Yeah, okay. Let's try room 3.
We didn't have much better. I wasn't sure.
Oh, okay.
Yeah.
Oh, how about room 4? Desiree, Tiffany, or Jude, did you guys have any answer for this?
No, we didn't get that far. We were stuck on that too.
Back on that one. Oh, okay. So it looks like this was a hard question. How about room 1? Kat, Sam T, and Jang. Did you guys have an answer for this one?
Yeah, we said blue-green algae.
Oh, okay. And why?
Well, we were looking at the graphs in the textbook,
Mm.
and it was saying, it was saying a lot of words, basically. But one of the things that they're talking about were controls and exclosures, and how in the absence of grazers, the diatoms are dominant, and then when there are grazers around, then the blue-green algal epithets are there.
Yeah. So if you've got colonial blue-greens, make it some great big blobby shape, that's really hard for a zooplankton to eat it. Or filaments that are all sticking together, that's going to be hard for them to eat it. So I would like you to, so I think the key would be when there's a lot of zooplankton grazers and not a lot of herbivores, then what you're going to get is the kind of algae that the herbivores can't eat, right? You put too many cattle on the grassland, on the prairie, and what do you get? Sagebrush. Because cattle don't eat sagebrush. They eat all the grass. If you put too many herbivores in an ecosystem, they will eat everything edible. And what do you have left? The stuff that's not edible. Big thorny shrubs, if it's grassland. If it's algae, you're talking about big blobby, inedible blue-green algae, big, hard-to-eat dinoflagellates, maybe big colonial green things. Yeah. Diatoms, you'd think they're made of glass, they'd be hard to eat, but actually, because of all of the pores in them, they're actually very crunchy, fragile. And in fact, I've heard diatoms described as being kind of like potato chips because for an alga, they're crunchy and oily. Yeah. Okay, so I would like you to, ponder this question of the ecology of algae, which this question begins, and this question kind of continues. And think about this in preparation for the next time we have lecture, which will be Tuesday after spring break. The graphs in this textbook that we'll talk about include this one, this important exclosure study showing how does the algae community change when you exclude grazers. And data like this that show how does the phytoplankton community change when when you change the amount of phosphorus in the water. If you put a lot of phosphorus in the water, you make it eutrophic. Very little phosphorus, you make it oligotrophic. How does that change the phytoplankton community? And it does in a big picture kind of way. And the question we really want to get to is why is it that at different times of year you see different types of algae? In the spring, lakes tend to be dominated by diatoms. In the middle of the summer, it tends to be blue-greens or maybe dinoflagellates. Why is that? What's the annual pattern of algae in lakes, of phytoplankton in lakes, and what causes it? So those are the kinds of ecology questions we're going to address after spring break. So I'll just introduce those things as something for you to think about. And that's what I have for you today. I will see everyone at the lake on Thursday at 1 o'clock, Lake Phelan, the same place we've been meeting. The ice will be fine. The ice is going to be totally fine. We'll go out on the ice. We're going to use triangulation. We're going to practice tying knots. It's going to be a nice day, 40 degrees or so. It is a little bit cold, so I think I recommend that you practice tying knots at home where it's warm, but we'll practice tying knots there too. I think that's everything. The lab is posted, and we will collect water samples and lab data that we will use in labs 7 and 8. Lab 7 is about lake ice and ice out and heat, or vice versa, heat, ice, and ice out. And lab 8 is a 2-week lab that focuses on Meromixis. And hopefully that will all go smoothly. And that's what we'll do. Yeah, that's my plan so far. Lab 6 is posted, 7 and 8 are not yet posted, but you just do what I tell you on Thursday and you'll be fine. Other questions? Any questions? We're all good? James says no. Oh, that's the no questions. That's not the all good. Okay, well, all good. You can give me a thumbs up, show me you're still alive. I promise next time we'll have sort of less lecture and a little more discussion, but yeah, we're all good. Okay, I will see you all on Thursday. Thanks for coming. It wouldn't be a class without you.